Hi guys, so in today's video I'm going to be continuing on from my last CAR T-cell therapy video, so if you haven't watched that yet make sure you go and check it out first. And in today's video I want to highlight some of the key challenges of CAR T-cell therapies. So as I mentioned last time, the most common type of CAR T-cell to date are CARs that recognise CD19 found on the surface of cancer B cells. However, not all patients respond to this strategy and many scientists are worried about the long-term control, as cancer cells are often known to become resistant to drugs after long periods of time and patients may end up relapsing. So one of the ways to try and beat this is to create dual-targeted CAR T-cells. So you engineer the T-cell to recognise additional targets on the cancer cells. And one example of this are dual-targeted CARs that recognise both CD19 and CD22, which is another highly expressed protein found on cancer B cells. And you produce CD19, CD22 CARs. And the idea is that targeting two molecules will significantly decrease the chance of the B cell escaping the therapy. And this is something most researchers take into consideration now when they're designing CAR T cells. Another challenge, which I think is probably the most important one, is how we can use these therapies in solid tumours. So for blood cancers, they've proven very effective, but that's because it's quite easy for the T cells to get into the patient's circulation and attack the B cells. It's much harder to get these cells to work in the case of solid tumours, and this is for a number of reasons. So firstly, designing an appropriate delivery method. So unlike in the blood, where a patient would receive an IV drip containing the CAR T cells, in a solid tumour, it's much harder for the T cells to reach the tumour sites, so it's far more likely that these will need to be injected directly into the tumour sites rather than just infused into the patient. Now, secondly, solid tumours are extremely heterogeneous, which means that they're made up of lots of different cell types, and not all the cells express the same antigens on their surface. And many cells are often hidden deep in the tumour, so designing a CAR T cell to target all the cells is extremely different. And there's not really any perfect antigen to target. And finally, in my opinion, the biggest challenge to overcome is the tumour microenvironment, which is often extremely immunosuppressive and very hostile. So as I mentioned in one of my previous immunotherapy videos, tumour cells express checkpoint inhibitors such as PD-1, and these can block the effect of CAR T cells, which eventually leads to CAR T cell exhaustion, meaning that the CAR T cells, even if they get to the site of the tumour, are no longer actually functioning. So now, while all of this is challenging, scientists aren't giving up and they're working extremely hard to try and combat all these problems. So some of the possible solutions, well, you've got DNA and RNA sequencing, which is being used to try and discover new neoantigens. So neoantigens are mutated antigens that are found only on cancer cells. And because solid tumours are heterogeneous, the use of tandem or dual CARs, as I mentioned before, will be crucial. So we can't target a single antigen, it will need to be multiple different antigens. And then finally, CAR T-cell therapies used in combination with anti-PD-1 therapies that I've talked about previously are also likely to prevent the cells from becoming exhausted and therefore be much more effective than just CAR T-cell therapies alone. Now, scientists are working on more solutions, but I think for the purpose of this video, I'll stop there. But I have included some useful links in the description if you want to do some more reading. So I hope you enjoyed today's video. And if you liked the video, please make sure to press the like button and subscribe to my channel to get notified of any future videos. Thank you.